I think my voice good enough, eh? Next speaker, Dr. Zamani from our, where is, yes, he's my, from the center, and he is our young colleague, and we have a lot of expectation from him. So first presentation was analytical. Now you'll hear a little bit about organic chemistry. So we choose in such a way analytical, organic, and physical chemistry. So hand over to you.
Good afternoon, everyone. As Professor Suprakas has introduced me, my name is Zamani, Cele. I'm working at the CSIR. So today I'll be sharing some of the work we've been doing on the chemical modification of biopolymers. So this work was planned in order to address the issue of antimicrobial resistance. So antimicrobial resistance can be defined as the ability of microbes such as bacteria, viruses, to evolve certain mechanisms that enable them to defend themselves against the treatment such as antibiotics. So, what is the crisis that we're actually trying to address? There is a big problem when it comes to antimicrobial resistance. The first thing is the lack of good drugs that are coming through. And the second thing is the research when it comes to developing these drugs is very challenging. And there was a report that was commissioned by the UK Prime Minister in 2016, where they were looking at the global impact of antimicrobial resistance. They stated that by 2050, if we don't address this issue, there will be 10 million deaths per year at a cost of 66 trillion. So one of the biggest things that compounded this issue of microbial resistance is the current COVID pandemic. So what happened is that recently, when patients were treating, when doctors were treating patients, they were using a same year where resistance was seen. So what we are trying to do is to try and address this issue by using biopolymers. So biopolymers, they are materials that are produced by microorganisms in nature. They are biodegradable, most of them. They have a lot of applications that we can try when it comes to biomedicine. So one of the most interesting about biomaterials is that as compared to small molecules, the resistance has not been seen, and also they have a multimode of antimicrobial action, so they can kill the bacteria using different ways. So in the past couple of years, we were more focused on chitosan. Chitosan is a biopolymer that is derived from cell wall of crustaceans. It has an amino group, and it also has a primary alcohol here. So we try and do modification on these two groups. So when chitosan is produced, it is produced in the form of chitin. Chitin is a deacylated form. So when we do base treatment, we form chitosan. It has a lot of interesting properties. It has some innate antimicrobial properties. It's biocompatible. It can be easily degraded, and it is non-toxic. And you can also control a lot of parameters, such as the molecular weight, the chain length, the charge distribution, and so forth. So what we did, we tried to modify chitosan. Here, I'm showing a little bit of chemistry, some of the work that we did. When you look at chitosan, these two groups, they can react, but the amine is normally more reactive than the alcohol. So we're trying to introduce some functional groups on the alcohol. So we had to block the amine using this phthalimine to form the phthalimide group. And then we were able to introduce some functional groups using allyl bromide on the alcohol. Then we deprotect the amine and introduce some quaternary functional groups. The quaternary functional groups, one of the biggest problems about chitosan is the water solubility. But when you introduce these quaternary functional groups, it becomes soluble. And when you do this modification, the chitosan itself changes and become nano. Normally, the size is around micron, above 1,000 nano, but after you do the modification, you get sizes that ranges from 100 to 500. So we then tested these materials against some bacterial strains. These were clinical strains that were isolated from the hospital. So what we saw is that you can increase the activity, and we also compare with the current antibiotics that are on the market. You can see here, when the chitosan is not modified, the activity is very high, but once you do the modification, you can see that they work against these bacterial strains. So we concluded that in the introduction of cationic groups, this one, and some hydrophobic moieties, this one, they can enhance the antibacterial activity of chitosan. We also did some more modifications where we introduced fluorines. Fluorine groups are used a lot in drug discovery. They enable the drugs to be stable, and they also offer some antibacterial activity. So we did these modifications, we confirmed them by the NMR, and we also check the degree of substitution, which tells us how much of the substitution was made on the amino group. Once again, we also introduce the quaternary amino groups to make them soluble and to enhance the antibacterial activity. We also did some titration to also check what is the degree of quaternization. And then when we did some antibiological assays, we found that these modified chitosans, they also show some activity, and we also did some kinetic studies, which shows that after 1 hour, you can start and see the activity, and after about 4 hours, they completely destroyed the virus. So in 2019, in 2020, when COVID hit, we were tasked to also respond to the pandemic. So we decided to synthesize more and more of these modified polymers, and then we screened them and tested them against the SARS-CoV-2, which was a pseudovirus. And when we did the screening, we discovered that the same quaternary chitosan does work against SARS-CoV-2. So what we did after we screened them, we immobilized the polymer into the mask, the inner part of the mask, and then we studied the percentage inhibition. So from here, A1 to A3, this is the mask that is not immobilized with the quaternary chitosan, and from here, this is the mask that is immobilized with the polymer. You can see that we managed to achieve 100% inhibition when compared to the unmodified mask, which was 50%. So these materials, they are very good when it comes to capturing virus, not just only bacteria, so they work against viruses as well. So in this diagram, I just wanted to show how we can apply these materials. So here I'm showing it can be any microbe, viruses, fungi. They can attach into a lot of surfaces, such as gloves, masks, and then even the medical equipment. So if you have, let's say, your coating here, which is a surface, if your coating is antimicrobial, the microbe, when it attaches, it will be killed. If it's not, it can still resist and be transferred and cause more infections. So these surfaces, they can be in the form of textiles, paints, antimicrobial paints, or food packaging and other applications. Their mode of action, you can have the surface that is completely antimicrobial. That is what we're trying to do. And also you can have drug delivery system where you have your drug incorporated into the surface and it releases over time. You can also have anti-fouling coatings. So still on chitosan, I'm just showing here some of the modifications that we did. Here we're trying to make amphiphilic chitosan. So amphiphilic means it has hydrophobic chain, and polar hydrophilic chain here. So what we did to introduce lauryl group, which is a long hydrophobic chain, and we also used succinic acid here to make this modification. So this material, when you put it inside water, it's self-assembly into like a micelle, because the hydrophobic, they point outside, and then the hydrophilic chains. So you get these micelles that are negatively charged. They can be used in drug delivery system. And when you do this modification, you can see that the particle size changes. This is the unmodified cytosine, and here you can see different particle sizes. And one of other platforms that we're working on is what we called Polymer Therapeutics, which is used in nanomedicine. So Polymer Therapeutics is when you try to solve the problems that are currently being faced with a lot of drugs, especially when it comes to pharmacokinetics, things like poor solubility, short half-life, toxicity. So basically what you do, you take a polymer that is non-toxic and that is soluble, that can be biodegraded as well. You combine it via any chemistry linker, then you achieve a drug that is soluble with increased half-life and also reduced toxicity. So we did a project that was funded by the World Organization. It was on malaria. So malaria is still a big problem, especially in Africa, South Asia, and South America. So in that project, one of the drugs that is used for the treatment of malaria is called lumefantrine. It's a drug that is not soluble in water, so it's normally taken orally in high doses. So there is a clinical challenge there where there is no combination of a suitable IV treatment for malaria. So in that project, we were tasked with a challenge of making lumefantrine water soluble. So what we did is that we used our Polymer Therapeutics platform, where we took a water soluble drug. This is the lumefantrine, the structure of lumefantrine. So we did a coupling reaction using coupling reagent. We reacted the alcohol and the acid to form this water soluble polymer lumefantrine conjugates. And when you check the size, it was around nanometers. So from this work, we characterized this conjugates using the FTIR, NMR. We also did some mass spectrometry. And from there, what we looked at is that lumefantrine is a yellow insoluble drug. When you mix it with water and you centrifuge, you check the supernatant, it's still clear. And our polymer conjugates, when you mix it with water, you don't even need to centrifuge. You get this clear yellow solution that can be used as an IV treatment. This one still cannot be used because it's like water solubility. From these results, when we did pharmacokinetic studies, we injected our polymer conjugates into healthy mice, and then we see a rapid release of lumefantrine in the form of liver metabolites, which is butyl lumefantrine. And then we also did some efficacy studies using infected mice. We observed a 50% reduction in parasitemia when comparing our conjugate to the lumefantrine that was not conjugated. So this is a very, very interesting result. And we believe that by taking this formulation and maybe further reformulating it and using other IV formulation, we can have a single cure anti-malaria drug that can be used as an IV formulation. And I think, yeah, that was my final slide. So in my conclusion, I would like to say that the reason we do this modification is that we want to improve the properties of these biopolymers. These are good polymers in terms of sustainability because they are produced by microorganisms in large scale, and they are very good for the environment. And also, it is very important to understand the mechanism of actions and structure activity properties of these polymers. So what we are currently doing, we are looking at things like computer studies, computational models, and also developing other bioassays to understand the mechanism of actions on how these biopolymers work. And lastly, the question can be asked, then what is next after you do all these modifications? So what I can say, we're also trying to work on seeing how we can apply this on solving real life problems. So we are making things like fibers, we're making things like hydrogels and films that can be used in clinical settings, and to try and see if they can help to reduce the burden on the antimicrobials that are currently used in clinics. Okay, this is my final slide. I would like to acknowledge all the companies that I've mentioned here and my CSIR colleagues. I couldn't list all of them, so I just wrote 2 clusters there. Thank you.
Thank you, Zamani, for showing that organic chemistry is a powerful tool to develop nanomaterials for practical application.
Yes.
So we have a time for one quick question.
And did you done any studies on the penetration, cytosine penetration?
No, not yet. We recently found some new collaborators at the University of Pretoria. They will be working on that.
Okay, thank you.
Yeah.
Okay, there is one, there is two. Okay, there.
Thank you. Hi. Sorry, I'm over here. Thank you so much. It was really interesting. I'm really interested about your mask work. Just in terms of like what kind of mask did you use, and has that been rolled out at all? And like what's the feasibility in terms of cost? That chitosan like coating that inhibited the virus or whatever it did.
I think we did some simple electrospraying where we take our polymers, we put it in solution, and then we spray it on top of the mask, and then we do some assays there to see if the mask can penetrate. Yeah, if the virus can penetrate.
Is it like a mask that I would wear, or is it like a CN95 medical mask that you used? Because it's just interesting, like if you could give that to a medical professional in a clinic, then they wouldn't be spreading. And so I'm just curious about that sort of.
It's the normal surgical mask. So what we do, it has 3 layers. There is the outer layer and the layer in the middle. So we open it up and then we coat the middle layer, so that we don't expose the polymer into you or the outer surface. Yeah. So it's the inner parts of the mask.
And how's that going? Like, where are you taking that? Because that's really cool work. Sorry, I just think that's awesome. I'm really just here to encourage you because I think that's fascinating.
Yeah, it's still under development.
Okay, cool.
The joint development with the SMME.
Okay. Thank you very much.
Transfer the technology, then now they are in a commercialized. Okay, go ahead.
Okay, thank you very much. Quite an interesting research. So I was like, did you look at other like biopolymers like alginate, which has some interesting properties? That's my question.
Yes. So we have other colleagues that are looking into that, because I think there is a lot of these biopolymers, and we like to explore all of them. But you can't do everything at once. But yeah.
Okay, so thanks, because of constraint of time.